Log on, tune in, find out. Another good idea from Cambridge.
Well, it's a great privilege to introduce John Bryant, who is very distinguished Professor Emeritus at Exeter University in cell and molecular biology. He's a visiting professor in molecular biology at West Virginia State University, where he's still running research.
No, I've stopped doing that now.
You've stopped doing it, okay. And he's a past president of the Society for Experimental Biology. He's given the London Lectures in Contemporary Christianity, and he's a past chair of Christians in Science. He is a member also elected in 2007 of the International Society for Science and Religion. He's a pioneer of education for scientists in bioethics. Started doing that, first did it, I think, in Britain in
1995.
1995, so that's been going for some while, and John helps with that here in Cambridge too. He's twice been listed for the National Award in Science Communication as well, so it's a great privilege to have you back with us, John.
Thank you, Ronnie. As always, your introductions are a hard act to follow. Now, before I start the substance, one or two people have asked me if I could recommend some sort of what I call getting into the subject books about the science-faith dialogue. And these are books that I find very straightforward and good to read. Unnatural Enemies by Kirsten Burkett, an Australian, sets out the kind of basics of why science and faith are not at loggerheads. Beyond Belief, written by Dennis Alexander and Bob White here in Cambridge. Science, Faith and Ethical Challenges. That overlaps a little bit with my bioethical interest, but that's a good one. And then John Pockingham. John Pockingham is a great writer, but some people find that you have to have a finger under every word. But this one, Science and Theology, an Introduction, is probably the most straightforward of his books, introducing. And then the book they produced last year, he produced with Nicholas Beale, a journalist, Questions of Truth. If you want to get into the science-religion interface, then I think one of these four would be very helpful to you. There are deeper tomes on the bookshelf as well, bookstand as well, if you want to do that. But these are good for introductions. Okay, now we're moving, if you like, in this talk from the basics of the science-faith dialogue into the way that we apply science and looking from a religious perspective, actually in my case a specifically Christian perspective, on what we can do with scientific stuff, the findings of science, and obviously my particular area is biomedical. So genetic engineering, how far should we go? I haven't got the U up there, ac.uk, this should be the last bit. If you want to get in touch with me, there's an email, but it's ac.uk, not ac.k, obviously. We'll do it. And I'm very happy to correspond and even visit if you think that would be helpful. Now, I want to say I'm using the term genetic engineering in a very broad sense, not just in the sense of moving genes, but in the sense of selecting genes. Engineering, if you like, being a kind of overall term of how we might use different methods to manipulate the makeup of a future human being, which includes direct genetic modification. So that's j.a.bryant@exeter.ac.uk. Now, I think we need to say that it's very, very easy to come up with black and white answers to questions which don't have black and white answers. You know, as famous management scientist once said, to every difficult and complex question, there's a simple and straightforward answer, and it's nearly always wrong. The straightforward questions the straightforward answers to these questions are often shallow and trivial. These are difficult questions. And they're questions about how individuals work out their responses to difficult situations. They involve families making tough decisions about family planning, about the future of their children and so on. These are difficult questions. And I'm very much working along the lines that we need Christian character, Christian virtue to be applied to these questions. And first and secondly, that different Christians will come up with different answers. And we should respect that. And I'm very much a communitarian in my idea of how Christianity works, that within the Christian community we respect the way that each other arrives at decisions. I should say a little bit more that my own spirituality is charismatic but very strongly influenced by Franciscan and Celtic thinking, so hence a big focus on the creation that you've probably picked up on the way through, and therefore reflective, very much a reflective spirituality. And this isn't easy, and I just love this. May God bless you with discomfort for easy answers and half-truths. I was also reminded, I was at a conference at Methodist Central Hall a few months ago, and if you go in there, there's a big poster that says, "May the peace of the Lord profoundly disturb you." And I think the same is here, that let's not have trite answers and assume there is a Christian viewpoint on these matters. So, I'm going to be exploring a little bit about our genetic makeup, what our genes are, what they do. That's the kind of science bit. And then I'm thinking about how we might select particular genotypes. And then I'm going to think about how we might directly modify genotypes by moving genes around. And I'm going to also give us some thoughts on eugenics. And if there's a moment towards the end, and I'm keeping an eye on the clock, then I might just briefly mention the idea, the practice of moving genes in and out of other animals other than humans. So those are the kind of threads that you'll see. So let's start with our genetic makeup and think about genes and DNA. As I said yesterday, DNA is a wonderful molecule. Without it, we would not be here. And it is my favourite molecule, and perhaps everybody should have a favourite molecule. For some it may be caffeine, others it might be ethanol, but mine is definitely DNA. And so, if we think of DNA as a string, not in the sort of string that Ronnie's been talking about in 27 dimensions, just a string with 2 nanometers across. As we go down that string, we can see individual tracts of it which have a job to do, an instruction, a recipe, and that is a gene. A gene is a specific piece of DNA that gives a specific instruction or provides a specific recipe, however you like to put it, for the cell. And genes are arranged in long strings called chromosomes, and we'll think a little bit more about chromosomes in a moment. So that's the kind of scientific basis. I think there are some handouts that could go round, actually. We've got all this on. Oh, here they are. So, and this is the heaviest science because it is going to get, I promise. So just imagine at the top there, where's my pointer gone? Anyway, at the top here we've got a piece of DNA, and within it we've got one of these bits of it that is a gene. And the gene is provided with punctuation. So it has places where the cell knows where to start reading it and knows where to stop reading it. It's like a sentence in that sense. And it's also got, at one end of it, part of the chromosome, part of the DNA, that enables the cell to switch the gene on and off. And that's a very, very important part of biology. Without genes being able to be switched on and off, we could not have multicellular organisms. So, the genes that are operating in my liver to whatever they're doing, aid my digestion and so on, are not operating in my brain. Although a subset of genes is operating in both types of cells. So genes can be switched on and off. The gene will provide a working copy of itself, which is called a messenger. And that goes out into the cell, and the messenger, the message, the recipe in that messenger is read by the cell, and it makes a working molecule, which we call a protein. So basically, the work of cells is mostly done by proteins, and the job of the gene is to provide the recipe through its messenger for the cell to make the protein. And that's, the science is going to get no more heavy than that. But we just need to keep that in the back of our mind much later in the talk, when we think a little bit about genetic modification, actually moving genes between organisms. And the recipe in DNA is coded, and it's coded simply by the order of the building blocks. The building blocks are called in common parlance bases, but they're actually really called nucleotides. And I just want to imagine those very cheap plastic necklaces that you used to be able to buy in Woolworths when it existed, when you can pop it together, little beads, and end on end on end. And DNA is really quite like that. The building blocks do pop onto each other, obviously in a biochemical way, to give us a very long thread. And there are only 4 of them, So imagine building a long thread of poppet beads just using 4 colours. And that you've got DNA. And the order in which those 4 bases are put together, which it obviously can be infinitely variable because the molecules are so long, provides us with our code. And the code is read in 3s, and those of you who have any biological training will recognise that I'm right at the start of a coding sequence there. The ATG provides the start of the coding sequence. And here's the fact that Richard Dawkins and David Attenborough shared in The Guardian last Saturday. Life is a unity. Every single living thing on the planet that we know about, and I think we've got representatives of all known groups, use the code in exactly the same way. So the bacteria that live in my gut, and me, we read the code the same way. We read the language in exactly the same way. And that just makes the hair on the back of your neck stand up. The first cell used the code this way. And we are descended from those first cells by evolution. So let's carry on thinking about genes a little bit. I've talked about them as instructions or recipes. Genes are therefore a coded recipe. It contains the instructions to make something, like a recipe contains the instructions to make cake or chicken chasseur or whatever it happens to be. And here we have these linear structures called chromosomes. And so all the genes on one chromosome are like a recipe book. And we've got 23 pairs of recipe books in our normal cells. And so, if you like, our genome, that is the collection of genes, is like a library of 23 recipe books. And we've got two sets of recipe books because we inherited one from mum, one from dad. So just think of genes as recipes. And the cell quite literally assembles from the messenger, reads the recipe, the instruction, assembles the protein according to what the messenger is telling it to do. Gets it out of the cellular store cupboards. Okay, so that's what genes do, and they code for these working molecules called proteins, but how much of us as human beings, how much of us, me as a person, do they code for? And Richard Dawkins would have us believe that even organisms as complex as ourselves have only evolved to be carriers of DNA, which is an extraordinary thing. And why do you make something so complicated? Just to be a carrier of DNA. And here's a quote from his book, River Out of Eden. DNA neither cares nor knows, it just is, and we dance to its music. So it's not just the universe as a brute fact, DNA is also a brute fact. We dance to its music. So the true utility function of life is DNA survival. But DNA is not floating free, it's locked up in living bodies and it's made most of the levers of car at its dispersal. Now, as a biologist, I found this an extraordinary saying, because actually, if you were just here to replicate DNA, you wouldn't bother to make multicellular organisms with 70 million cells in them. You'd just stick at bacteria. And so it then says the genes, this is my interpretation of the term selfish genes. People misunderstand the term selfish gene and think it means a gene for selfishness. And doubtless, that our use of our genetic freedom, our intellectual freedom, can make us selfish. But Richard Dawkins does not mean genes for selfishness. He means the genes are selfish. That they are there to replicate themselves and nothing else matters. So that's his view. It's not a view shared by many biologists, incidentally. So the question is then, how much of our other interests and activities are built into our genetic makeup? You won't recognise the team I support because they're in their away strip there, but they are at the top left. Those of you who know that I come from South London will recognise the, or you can guess at the team I'm supporting. This is at St Mary's Stadium in Southampton, and Southampton are the home team, therefore they're in their red and white stripes. Okay, but what about sporting prowess? I feel sure that Beckham's little boys will be quite good footballers. Is that nurture or nature? And here's my hero, this is Paula, and she's now pregnant with her second, and those children will doubtless be runners, just as my 2 sons are runners. And then this is a young woman who has just graduated in jazz at Leeds. She's a member of our church, a friend of mine, and she is a remarkable talent, already has her own band, which is making money, her own jazz band, and she's the lead singer. You know, how much of her talent comes from her genetics? These are the sorts of questions that relate to what we might do with genes, what we might do with human beings in the future. How much of our, if you like, the non-basic physicality of nature is in our genes? Sporting talent, musical talent, poetry, and so on. It's a big question. And I put this one up because it's such a lovely picture. One of the things we have done at Exeter, which was set up during my time of kind of running the department, was to set up a year abroad for biology students, mostly in English-speaking countries. And one of the places that I set up an exchange with was the University of South Carolina at Conway near Myrtle Beach, which is kind of Sin City, the East Coast. But this is part of the cheerleading team for the American football side they have there. And what I just love about human genetics, which has come up from the Human Genome Project, is the similarity. It's not just we all share the same genetic code, we're actually quite similar genetically. So the diversity that I see in front of me is actually based on quite small amount of actual genetic variation. And as I said before, if I looked at the genes of a black brother, he and I would be pretty similar. And so if we look at these two young women here, the black girl and the white girl are probably as genetically similar as the two white girls. You know, race is very superficial, and it has all to do with our evolutionary past and being adapted to particular climates and environments. So it's an extremely superficial thing. Now, as for this here, that's some sort of mutant. Clearly, some sort of genetic change is taking place. Those of us who are bird watchers find this a very difficult specimen to identify. The team are called the Chanticleers, and that, believe it or not, is South Carolina's version of a cockerel, the Chanticleer in Chaucer. It looks to me more, it's got a lot of raptor about it. So that's their mascot. Anyway, let's move on. We've thought about things like sporting prowess, and clearly we can identify genetic threads that relate to ball control, to hand-eye coordination, and so on. But what about behaviour and personality? Well, I think the first thing we need to say is that our brains are the physical entities through which our personality and our emotions and our thoughts are expressed. And genes obviously regulate the biochemistry and the physicality of the brains. Without genes, we wouldn't have those structures. And so the molecules and the electrical signals that conduct in the brain are based in our genetics. And we now know from careful and detailed inheritance studies in large groups that aspects of behaviour and personality are heritable. And I'm not going to put a percentage on it because it's very difficult to do that. You know, that we're 50% nurture and 50% nature. We can't do that. But what we can definitely say is that there are elements of our personality and behaviour which we can detect as familial traits, which have nothing to do with living in the same home. They are part of our genetic makeup. So putting a finger on the amount and how that works, we're still very far from doing. The other thing I want to say is that genes are not everything, even in terms of the physical makeup of the brain. We're finding more and more that, if you like, the emotional environment in which a small child is raised, particularly in the very early months and years, going up to about 2, affects the way the brain works. And some of you will be familiar with those very, very sad pictures that came from Romania after the Ceausescu regime was overthrown, of the babies who've been abandoned in orphanages with no emotional and physical comfort. And the brain scans showed quite extraordinary differences between a normal brain and a child that's been extremely neglected. There are huge lacunae, holes where there shouldn't be, and we're beginning to see some necrosis and withering away of cells. But severe emotional deprivation has therefore affected the way that genes work. It's not just the physical environment that affects the way that genes work, but the emotional environment can affect it too. My wife works with families in deprived areas of Exeter, and even in a middle-class city like Exeter, there are some aspects of emotional deprivation that we see in children in Britain, where the lesions are not nearly so marked, but nevertheless they are there. And you have only got a very small window in order for that to be repaired by restoring some sort of emotional care and comfort for those children before those set in permanently, and then linguistic development is permanently impaired, for example. So genes are affected by environment, and that environment includes our emotional environment, which is an extraordinary thing. We are very much whole people. Okay, let's then move on. One of the things that drove the Human Genome Project was the understanding that genetics has got a lot to do with illness, and it's got a lot to do with illness in at least two ways. Firstly, there are those occasions when in the life of an individual a gene stops working properly, or a set of genes stops working properly. And that's within the life of an individual, not necessarily inherited. So some forms of cancer would fall into that category. There are also a specific number of diseases where we can trace an inheritance of the condition. So there's been a change in the gene, which is passed on from generation to generation, where we can trace a 100% linkage between the condition, the illness, and the gene change. And so there are about 4,500 of these diseases, single gene disorders. And I've already said that gene activity is affected by emotional environment and by physical environment, but nevertheless, we've got this clear-cut linkage between genetic change and mutation and the presence of a disease state. Now, I would say that in some of those cases, the disease state doesn't develop in everybody that's got the mutation. So, for example, in the breast cancer mutations, about 70 or 80% of the carriers of the mutation develop the condition. But nevertheless, it is a specific direct linkage between a gene change and an illness. Colon cancer, that percentage is 60 to 70. We call that penetrance, 60 to 70% penetrance. In other instances, if you've got the genetic change, then 100% certain you're going to get the condition. But these are direct linkages between a single gene and a disorder. But in addition to that, we are beginning to understand that genetics comes into a lot of ordinary illnesses. And so, for example, you may have a colleague you think is a bit of a malingerer and seems to stay at home whenever there's a flu bug about. But they may well be genetically more susceptible to virus infection. And the interaction between those susceptibility genes and somebody's environment is very strong. And if I think about Roy Castle, the trumpeter, who never smoked in his life, but he played a lot in clubs where there was a lot of smoke, and he died of lung cancer. Clearly his genetic makeup made him more susceptible to the environmental carcinogens. I have another friend who has smoked 60 a day since he was 17, and is now approaching 70 in apparently good health, except he coughs a lot. And he's clearly genetically not susceptible to the carcinogens in cigarette smoke. So susceptibility is going to be very much part of modern genetic medicine. So here's some examples, and I'm using these examples to set the scene for what I call difficult decision making. We have the recessive illnesses. In terms of the serious conditions, cystic fibrosis is by far the commonest condition in the Caucasian populations of Western Europe. There is another liver condition which is commoner but is much, much more readily treatable. Cystic fibrosis is effectively untreatable, you just keep it at bay. And by recessive illness, I simply mean an illness where you need both copies of the gene, the one in your mum's set and the one in your dad's set, to be mutated in order to have the illness. If you've got one decent copy, it overrides the faulty copy. So those are recessive conditions. But as I say, cystic fibrosis is the commonest serious condition in white Europeans. The carrier frequency over in this part of Britain is about 1 in every 25 people. So the chances of meeting and having a child with another carrier are really quite high. And the further west you get, the nearer you get to the Celtic fringes, so the carrier frequency goes up. And so in Cornwall and in Brittany and in Wales, so if you're of Celtic stock as I am, the carrier frequency approaches 1 in 22. This is almost certainly what we call a Celtic founder effect. It arose in a small group, and the interbreeding has not yet had time to spread it at an even rate through the white populations. And here's a similar case with sickle cell anemia, which arose, we think, in West Africa and spread to North Africa and thence to the United States from West Africa because of the slave trade, which is why it has been so prevalent in African Americans. Sickle cell anemia, which affects the way that your cells carry oxygen around your system. So those are recessive. You need 2 faulty copies, 1 in each set, to have the condition. And then you have dominant conditions, Huntington's, and by dominant I mean that you only need 1 faulty copy and you're done. You don't, the good copy won't override the faulty copy. I'm putting up the code in DNA to remind us that this is what we're talking about. We're talking about changes in the genome that have led to the condition. And Huntington's is very strange, but Huntington's disease gets more and more active down through the generations. And so the first time the mutation occurs, it may be that it onsets at 50, 55. So people had their kids by then, and they don't know they've got the condition. Then it goes down, maybe in the next generation it's 40, 45. And the youngest sufferer of Huntington's that I know of was a 7-year-old child. And the reason it increases its severity or comes earlier and earlier is because it involves an insertion into a gene, not a change, but an insertion. And the number of those insertions increases through the generations. So the coding sequence is interrupted, and it's interrupted more and more as you go down through the generations. And it ends with a very nasty neurodegeneration. And basically the brain stops working the body. And people then die shortly after the onset, well, a few years after the onset. A nasty disease, quite prevalent in South Wales, for reasons we do not understand. And then there are X-linked diseases. These are recessive conditions, but they're carried on the X chromosome. And guys, if we've got a mutation on our X, we are buggered because we haven't got another X to override it. The Y chromosome has some important stuff on it, but it's not very big in terms of its genetic content. I mean, the important stuff is testis determining factor and football maybe, air guitar, this sort of thing. Heavy rock music. No, that's not true. Some good heavy rock female musicians, Chrissy Hynde, etc. So X-linked conditions then are conditions where the mutation is on the X chromosome and therefore men suffer from them. And haemophilia is the classic. It is Queen Victoria's gene. So she had 1 faulty copy, passed on the gene to 50% of her male offspring. And through the crown heads of Europe, you can see her gene going down through the European royal families. It was interesting, this was a mutation, an actual mutation that was discovered long before we knew anything about genes. And Steve Jones tells this story in his wonderful book, In the Blood, of a Jewish rabbi, during the Christian era actually, it was about 8200, a Jewish rabbi circumcised a little boy, classic, on the 8th day of the Jewish rite, and he bled to death. The wound didn't heal. And later on, that same couple had another little boy, and he was circumcised, and he bled to death. It was very sad, but the rabbi thought, there's something strange going on here. And so he said to the family, heard any one of the boys, we won't circumcise them, we'll welcome him into the Jewish faith without circumcision. And he said, we better do the same for the male cousins. Now, it was very wise of him. He had discovered, although he didn't know, X-linked haemophilia. Okay, so the thing that the Human Genome Project enables us to do is not just to say that we know that diseases are caused by changes in genes, that throughout the world, from the time we could start mapping genes and pinpointing where they were on chromosomes, and then determining what had gone wrong, all that started in the 1970s, but from the 90s onwards, it became a concerted effort across the world, in all the countries able to do this, to look at what was going wrong in these inherited conditions. And I appointed a young woman who has gone on to much higher things now. She's a professor in Newcastle, who was looking at diseases of blood groups based around chromosome 19 in terms of this sort of approach. We're beginning to be able to pinpoint what has gone wrong, as I described, for example, for Huntington's. And that's enabled us then to test for illnesses. And the thing about DNA is that it may not be as powerful as Richard Dawkins says it is, but nevertheless, DNA tests are themselves very powerful because you don't even need the patient to be in the surgery. You don't even need the symptoms to be apparent. You just get the DNA and see whether the genetic change has occurred. And we can test adults. And so, you know, some of if Huntington's occurs unexpectedly as a new mutation, and the doctor suspects, then you can test an adult. We can test children. Maybe an 11-year-old boy is developing muscular weakness. You can test to see whether that's caused by Duchenne muscular dystrophy. We can test newborns, and indeed we do test newborns for a handful of genetic diseases already. A small amount of blood is taken from the heel a couple of days after birth, and the number of genetic conditions that we routinely test for is about 5. It does depend on which community you come from, which you're tested for. For example, Greek Cypriots are routinely tested for an illness called thalassemia, which is another blood disorder arising in that part of the Mediterranean. We can test fetuses, and I'm looking forward to listening to John Wyatt this afternoon, who will talk about some really tough decisions when couples find that their fetus carries a severe abnormality. Some of these abnormalities are genetic. And we can test pre-implantation embryos, and this is what I really want to focus on in terms of the selecting our genetic makeup. That's been really quite a long introduction to the topic. So we're going to now work our way through this, actually working out what to do. IVF was invented in Britain. I think it's very salutary to know sometimes that science goes on behind lab doors without the public knowing. When I was a postgraduate here in the late 60s, in the building next to mine, there was this crazy man, Dr. Edwards, who was trying to grow mouse embryos in the test tube and then get fertilization. He thought, that will never work. 10 years later, the first human test tube baby was born, a petri dish baby, really, Louise Brown. Therefore, since 1978, we've been able to handle embryos in the lab, and we're getting better and better and better at it. And that has included, since the Human Genome Project and since the advent of the new genetics, that's included being able to genetically test those embryos. And the reason we can genetically test them is there a pointer somewhere? I thought there was one on the table.
I had it, didn't I?
It was there. Anyway, I'll wave my arm. The reason we can test mammalian embryos is because of the extraordinary way that mammals develop. Here we've got the first 7 days in the life of a prospective human. They are taking place in the petri dish. In vivo, in the living woman, they'd be taking place in the fallopian tube. So these are the pre-pregnancy stages, and I do mean pre-pregnancy. Even in life, a woman isn't pregnant. The pre-pregnancy stages of human development. And at the top, we've got a newly fertilized egg. The two sets of genetic material have not yet merged, and they won't merge for several hours yet. So we call that the zygote, and I like to talk about the process, calling it fertilization. I think the term conception here is extremely misleading. The term conception arises from an earlier era when we did not know that women produced eggs. So let's stick to the term fertilization for that early event. And for establishment of a pregnancy, we have to have this structure embedding itself into the wall of a womb. This is the blastocyst. I'm not going to talk any more about that. But for a woman to become pregnant, that actually must latch onto her womb lining. And it happens successfully in about 20 to 30% of fertilizations. So 70 to 80% of fertilized eggs, early embryos, don't actually establish pregnancy. So pre-pregnancy existence of human embryos. And the other thing about human embryos is that until this point, there is no decision making going on at molecular level about what the cells are going to do. At this point, we begin to see this will be the placenta, this row of cells, and this little blob of cells will eventually become the fetus. But at this point, which is about 2 days earlier, the cells have no idea what they're going to do, as it were. We don't know what they're going to do. And you can take one out, and the rest of the embryo says, oh, lost one. Well, we'll carry on. And extensive experiments on mice, including experiments here in Cambridge, showed that development went on normally as far as anybody can tell, if you took one cell out at the 8-cell stage. Now, DNA technology is so powerful that you need one cell to do genetic tests. Isn't that astonishing? One cell. And so this is what you do. This is quite an old picture where one cell is being teased out of an 8-cell embryo, about 2 to 3 days old after fertilization. And that cell is going to be used for genetic biopsy. These days, in the last 4 or 5 years, instead of teasing it out with needles and effectively micro-cutting instruments, we use a laser to slice the cell out. But it's exactly the same in principle. One cell is removed. And so we have pre-implantation genetic diagnosis. Now, supposing that you know from your family history that you are at risk of having a child with a severe genetic condition. Just think about the family situation. Would you start a pregnancy and then have the fetus tested? Or would you prefer to undergo IVF and have the embryo tested? Is one more ethically acceptable than the other? Some do indeed find pre-implantation genetic diagnosis much more acceptable than termination of pregnancy. I used to say this in theory, but I know a young couple actually facing this situation. And they say for them that disposing of embryos of faulty genes is much, much easier ethically for them than terminating a pregnancy. So that's what happens. The couples who are at high risk will attempt to select disease-free embryos. And also we're seeing that some couples use this technology to select for a saviour sibling, i.e. a sibling who will save an elder child. And I'm going to talk a bit about, I'm going to give you a specific case of that in a minute. And over here, in Britain, all these things are regulated by the Human Fertilisation and Embryology Authority, the current chair of which I think is Lisa Jardine, the history professor at University College. So here's some stories. These are all real cases. Matthew's story. Now we don't actually know the names here because this is a British case. And anonymity is protected unless the couple don't wish it. And this couple didn't wish it. This is the very first case, or maybe the second case, of pre-implantation genetic diagnosis in Britain in 1989. Helen and Peter knew they were carriers of the cystic fibrosis mutation to be in their families, and they had their genes tested. And they knew, but they didn't want to have a child with CF. It's an unpleasant condition. So they underwent IVF, embryos were tested, and only embryos free of the condition were implanted into the woman's uterus, into Helen's uterus. So these are made-up names. And Matthew was born healthy as far as the cystic fibrosis goes. When we talk about healthy, we are only talking about the gene in question. We will get other illnesses. So that was the first instance of a couple in Britain using pre-implantation diagnosis to have a child free from a genetic condition that they knew they were susceptible to. And in the same year, there was a case in the United States, but not for cystic fibrosis. Okay. That seems very straightforward, but I'm now going to kind of rack up, if you like, the ethical tension. This is an American story, and if you want to see the video on it, you know, the couple went public, and there's a nice video, www.nytimes.com, and put in the search box, embryo screening video. And the video is about 4 years old now, 3.5 years old, 4 years old perhaps. So here we're talking about colon cancer. A dominant mutation. If you've got one copy of the gene, then you have the illness, or you have the probability of having the illness. So in this particular case, and this is a real live American couple, remember, so they've got real live American names. So we have Chad, who's the dad, and Colby, who's the mum. And you can look at Chad's family history, and you can see, you can trace the occurrence of colon cancer. And most recently, when the video is made, one of his uncles has just died from the condition. Chad knows he's got the mutation, having been tested. Now, this is a late onset condition. And unlike cystic fibrosis, which is present from birth, this is late onset. And so, generally speaking, the first signs you have of this form of colon cancer occur during your 50s, that sort of time. And its penetrance in the States is only about 60%. for reasons we don't understand, the penetrance in Europe is about 70%. It's the same genetic mutation. So the penetrance is not 100%. So we've got a late onset condition which you may not get. And with modern medicine, if detected early enough, and in fact even with quite late detection these days, the cure rate for colon cancer is very high. I know 2 people who are well into years of remission after colon cancer treatment. So we've got an interesting state here, a rather different situation from maybe Matthew. But Ted and Colby decided to undergo IVF, and the video talks about their decision making, and it does examine the ethical background, and they didn't want a child with that mutation, and Chloe was born, and Chloe's about 6 now. And so that was an extension, if you like, of PGD into a slightly less clear-cut situation. And I'd like you, as we go through these cases, I'd like you to think about, if this was my family, what would I do? Because real ethics is about making decisions. My friend Susie Leather said when she was chair of the HFEA, it's no good ethically theorising. The couple needs to know whether they're allowed to do this. And we can philosophise for months, but it doesn't help them at all. So these are decisions that need to be made, and we're talking about, in this instance, within the Christian community, although these are not Christian people. And in fact, they talk about the objections of Chad's parents' church to what was going on. Okay, this is another real story, and this is again another from the States. This is baby Adam. So we have Molly Nash. She was born in the state of Colorado, but the technology wasn't available to the couple in that state, and so they went up to somewhere in the Midwest, somewhere like, I think it was Minnesota, to have the tests. Molly Nash is born with Fanconi anemia. It's an inherited condition, it's recessive, and it usually leads to death in the teens. However, like many blood disorders, if you can provide a donation of healthy stem cells from the bone marrow or from the umbilical cord, you can provide healthy stem cells, you can often cure the condition. And that is true of Fanconi anemia. And so Molly's parents elected to do just that, to have a baby. Remember, Molly was 7 at the time, they hadn't had another child. They had said they kept putting it off. But they elected to have another baby by IVF with pre-implantation genetic diagnosis. This is to generate what we call a saviour sibling, somebody who will provide cells to save somebody who's already here. And Adam was born, and the stem cells, as it happened, from his umbilical cord were enough to cure his elder sister. I will say that had they not been enough, then they would have called on his bone marrow itself when he was a little bit older. And Molly is now 15, and her mum says she's a normal teenager, hangs around the shopping mall on a Saturday, and texts her friends on her cell phone. Exciting life being an American teenager. She's unfortunately got juvenile onset diabetes, but that has nothing to do with her Fanconi anemia, so she doesn't feel she's had a good deal in life, but nevertheless, she gets on with it. Now, the downsides of that sort of donation are beautifully described in the novel by Jodi Picoult, My Sister's Keeper, where a girl feels she's just there to provide cells for her sister. Fascinating book. I was also involved in writing a play which has toured many universities and colleges called A Present for Anna, which is about decision making in relation to Fanconi anemia. Now here's the last one. This is a curious one. Tom, Paula and Molly. Tom, these are both theatrical folk, and Tom's professional name is Tomato. I don't think he was actually named that at birth, but he calls himself Tomato, but I'm calling him Tom because if you present this to sixth formers or fifth formers, they start giggling with Tomato. So they're both deaf, and they have a heritable form of deafness, and their first child, Molly, was born deaf. So she's born deaf into a deaf family. And they wanted to ensure that any other children they had were also deaf. And they applied firstly to their local genetics clinic, who refused, referred it up to the HFEA, who refused. The Human Fertilization and Embryology Authority refused on the grounds that the guidelines state that there should be no positive selection for conditions that are regarded as a disability. and deafness in the general community is regarded as a disability. Tom and Paula argue that in the deaf community, deafness is not a disability, and with British Sign Language being adopted, being recognised as a language, and American Sign Language being recognised as a language, then we've got a whole kind of community of deaf people. The application was rejected. They took it to the highest court they were allowed to take in Britain, still rejected. And that's how it stands at the minute. They've not been allowed to select for a deaf child. And that's something that you might want to ponder. Okay, so that's genetic testing pre-implantation. The technology is growing. The data on your handout are one week older than the data here. So we've got, across the world, we've got over 1,900 genetic tests available. I will say straight away that they're not all brilliant, but some of them are not very informative. Some of them just talk about percentage probability of. The fact that many of them can be offered commercially by organisations with no deep medical backup means that sometimes the advice that goes with the positive test is very, very weak, even poor, even misleading. So the commercialisation of genetic tests is worrying us in the science community, that the tests often come without proper help, counselling and advice. And in Britain, it's been agreed that in only well-established tests, where there is a clear-cut answer, and which relate to serious conditions, shall be allowed to be used with embryos. And about 100 have been licensed. Hypercholesterolemia is elevated cholesterol, which even if you've got one copy of the faulty gene makes you pretty sick. If you've got two, you die. Late onset conditions, they will allow, like Huntington's, that seems reasonable. And predispositions to cancer, there are some quite well worked parts now, predispositions to cancer that we're beginning to pick up. So those sorts of things are allowed. So, what are the key ethical conditions, issues in the genetic testing membrane? Where for you are the ethical issues? And one of the ethical issues, there are issues relating to this, what is personal and is the embryo personal? But one of the ethical issues is how far. The talk is called genetic engineering. How far should we go? How far should we go in this form of genetic engineering, which is selecting our genetic makeup? Where are the boundaries? Should we step outside the realm of medicine, influencing behaviour, intelligence, sporting ability, or genes that affect our appearance, what we might call cosmetic? Some of these are genuinely possible. Within 5 years, we'll have an 80% chance of getting eye colour right. It's about 65-70% at the moment. And 80%, one clinician in San Francisco says we will offer that genetic test for couples starting families. So while I sit on top of this Monroe, you can ponder that. So just a little break to think about, what sort of thoughts does that raise in your mind? What we've been talking about. And feel free to talk to your neighbour or just ponder it. We'll just have a short break, a minute or less. What about actual modifying our genetic makeup? Here's a picture of some DNA molecules because I always have to show some. Modify our genetic makeup. Let's think of a newly fertilized egg. This is a mouse egg newly fertilized, and we can inject DNA into it. Mammalian, individual mammalians are rather curious that many of them would accept DNA that you add to them as if, oh, that looks good. And so with things of long generation time, like monkeys and humans, you would need to use the one-cell embryo as the target for putting new genes in. For short generation mammals, such as mice, there's a method that we can use stem cells for, but I won't go into that. But the thing I want to say is that it's completely and utterly feasible to genetically modify mammals. We know it is because we do it. And those mammals vary from cows down to the smallest mammals, the small rodents. So in GM of humans, we normally think of GM of humans in relation to correcting faulty genes. And for for those, we wouldn't actually use the newly fertilized egg. I'll tell you why in a minute. It's called somatic gene therapy. It applies to the body. And what we have to do is we have to find the cells that are most affected by the mutation. Ones which are, so for cystic fibrosis, we go for the lung lining. For immune deficiency diseases, we go for the bone marrow. For diseases of sight, we will go for the retina. So here we're not using the egg, we're using the cells that are affected by the disease. And there has been some success with cystic fibrosis, a very small level of success. You know, probably in terms of the number of cases that have been tried, it's probably relates somewhere in the order of 1 to 2%. But with the severe combined immunodeficiency, which means that the child is susceptible to every single thing that comes along, the success rate for curing the disease with the modern technology has been nearly 100%. In the two centres are Great Ormond Street and Institut Pasteur in Paris. The success rate has been nearly 100%. There has been a side effect with about 10 to 15% of the children who develop as a result of the genetic change. They develop a form of leukemia, but that's also been cured. And some of the early cases at Great Ormond Street are now in ordinary primary schools, you know, subject to all the buffeting and viral illnesses and childhood, this or that and the other, and are living normal lives. So for them, gene therapy, changing the faulty gene in their bone marrow has been a real blessing. And I think it's a rare disease, but it looks like if you have a child who's born with it, then changing the faulty gene will work. And then there's recently been congenital blindness, 2 cases now, very experimental, where a gene is being changed in the retina and people are beginning to see, one in the States, one in Britain. And more recently, faulty genes have been switched off to allow in dominant conditions, to allow the healthy copy to actually work. Faulty genes have been switched off by a technique which is called RNA inhibition. I'm not going to talk about the technology of that. So different forms of gene therapy are developing, but actually for most illnesses, it's impossible because you cannot get at the target cells. So it's a limited range. Now, there's also germline gene therapy, you know, correcting a genetic illness in the fertilized egg so that it is passed on to the next generation. What I've been talking about is that the genetic change would not be passed on to the next generation because the genetic mutation is still part of the makeup. But if we were to put it into the fertilized egg, every single cell in the future human being would have to change. It's heritable, therefore. But actually, in Britain, you're forbidden to do it. You must not put a genetically modified embryo into the womb, even if you've done experiments on those embryos up to the 14-day stage, which is permitted. You're not allowed to make a pregnancy. And yet, if you go out to the streets of Cambridge and put this scenario to them, I bet that there'd be a majority of people who think it would be a great thing if we could heritably cure genetic illness. My own view, actually, is that the call for it is very limited because, in fact, pre-implantation genetic diagnosis, much better established technology, is almost certainly the route that a couple would take if they knew that they were at risk of having an embryo with a genetic illness, rather than trying to genetically modify an embryo that had the genetic disease. But of course, if we went down, if we opened up the door to germline simply means it's passed on heritability. If we went down that road, then straight away we run into problems, because if you can do it to correct illness, you can also do it for heritable enhancement. You know, you can do it. And it's very hard when you look at the kind of range of possibilities. It's pretty hard when we're looking at genetic selection, pre-implantation diagnosis, sometimes to tell the difference between a medical and a non-medical. And it gets harder and harder as we think about the whole range of parameters that make up a human being. And the example I like to talk about in relation to telling the difference between therapy and enhancement is that there are some young women who are under the mistaken idea that enlarging their breast is an enhancement. But some of them argue that on therapeutic grounds, that, you know, their depression will be cured if they went up from an A cup to a C cup, or whatever it happens to be. And so they're arguing that this is therapy and not enhancement. And so you can find all sorts of cases in which surgery has been used not for medical cases, but for if you like, cosmetic cases. And I think the same will be true of genetics. It'll be very hard to tell the difference sometimes between therapy and enhancement, as we can see there. Where are the boundaries? Now, I will say that there are some people who think we should have open house, both on genetic modification and on genetic selection. There should be no guidelines. The HFA is being too restrictive. And thus, the atheist philosopher at Manchester, John Harris, says, if a couple wants a bouncy, curly-haired, brown-eyed girl, I don't know if he's a Van Morrison fan, why don't we let them have it, rather than having them rely on nature, and then he puts in brackets, or their God, giving it to them. Why don't we just ensure they can have it? So that's his view, open house. And there are certainly, Gregory Stock at UCLA, another policymaker in this area, is of exactly the same opinion over there in California. This raises the idea of designer babies then. Does a baby become a commodity? Well, some people have babies for all sorts of reasons already, save their marriage, fashion accessory, something to love. Now, I think let's be very clear about this, that both genetic selection and genetic modification require IVF. That has not become mass technology. We're not yet in the Brave New World scenario. It's still pretty difficult. And therefore, I think we need to beware the science fiction. So we need to be aware that these are possibilities, but we need to beware of the science fiction that are often presented, even in the serious media. Okay, so in the last few minutes, I just want to raise some thoughts on eugenics. The U here is the same as in euthanasia. It means good, and so this means good inheritance, good breeding. It doesn't mean to say you hold your knife and fork properly. It means you've got a good set of genes. And Darwin wondered, without thinking about it any further, whether evolutionary theory would work with human society. But actually, he was 2,000 years too late to be original, because Plato and Aristotle had also come up with that view, with their view of what the ideal community was like. I think it was Plato who suggested that the best men should have sexual intercourse with the best women as frequently as possible. He, of course, was defining what was good, who were the best, to generate the best in human society. And Francis Galton, the first professor of genetics in the UK, in fact formalised the theory of eugenics in a very similar way, that we needed to prevent the criminal underclass from overbreeding and to encourage the liberal middle classes to breed more. He was a bit worried that human society was degenerating because of the breeding habits of different groups. It makes huge assumptions about what's built into our genes, most of which are not correct. So let's encourage. And eugenics movements, we tend to think of Nazi Germany, but I think let's look at this lot. In the UK, it was never really formalised, but in some of these countries it was formalised in law, negative eugenics. 50,000 people in the States were sterilised for being morally enfeebled, and there's some very tragic stories about that. And the last eugenic sterilisation that we've been able to detect was in 1974 in Sweden, and I was a script advisor for a play about that. So, are we on a slippery slope here? Philosophically, ethically, I'm not a great believer in slippery slopes, but I think in the whole area of genetic manipulation of the human condition, I really do detect we are on a slippery slope because of this lack of a clear boundary between genuinely medical and therapeutic applications and less medical and then cosmetic and then further enhancing. It seems to me it's the one case where I think a slippery slope might apply. Okay, so we're going to change tack very slightly and to think about an overview. This is what Madeline Bunting says in The Guardian. She's not talking just about genetic selection, she's talking about a whole area called transhumanism, which we could spend an hour talking about as well. Interfacing humans with IT devices, drug implants to enhance activity. But the tools to make us cleverer and stronger. In genetic terms, she's talking about things like sporting prowess and that sort of thing. Resistance to illness. So there's no stop button in the race for human re-engineering. Her view is that we are on a slippery slope, and it's without an ice axe. In some way, we need to find a stop button. So let's very briefly then consider non-human animals. The genetic modification of mammals was developed initially on mice and very rapidly spread to farm animals. And one question I will throw out is that does this raise any ethical issues that are not raised, for example, by animal research in general, or is it a different set of issues? Are we offending against the animal nature of the animal, the telos, putting in genes from somewhere else? So that's something to think about. And the applications that we see actually working at the moment are in large farm animals. Agricultural features have tended not to work. Trying to modify body mass ratios tends not to work. Increasing milk yields by this technique tends not to work. But what does work is the ability of, in particular, sheep to produce pharmaceutical proteins in their milk. And for some genetic conditions, or some illnesses which require a protein as therapy, you can in a flock of ewes of maybe 40 or 50, you can have enough to supply the needs of one nation for a particular product. So pharmaceutical products in sheep's milk is seen as a very easy way of taking pharmaceuticals into countries where the kind of technology that's normally used for GM drugs, large fermenters, would be out of their range of possibilities. Developing countries, just milk the sheep and you've got your therapeutic product. Dolly is cloned not to cause a sensation, because the sheep from which she was cloned had been genetically modified and was producing a very valuable pharmaceutical. The genetic modification of farm animals has only about a 15% success rate, and so if you can clone them instead, you get lots of copies. But nevertheless, it is actually commercially viable. The largest number of genetically modified mammals, there are probably approaching 5 million in the UK at the moment. They're nearly all mice. And scaremongers will tell you, did you know there are 5 million GM animals in the UK? Yeah, but actually they're all in labs. And what people tend to do is they tend to, they study the development of human genetic disease by putting in human genes, disease-causing genes, into the mice, and thereby help develop treatment. So there are cystic fibrosis mice, for example. There are mice with breast cancer genes, and so on. And again, you may have issues about that. But as I said, there's probably about 5 million GM mice being used in labs right now. Some as breeding stock, some for actual experimentation. Then pigs, this is the most recent application. There's a huge, huge shortage of organs for transplant. The waiting list for organ transplant, particularly kidneys, but also for other organs, the lists are growing faster than the number of donors. So people are dying as they wait for organs. And so there's quite a lot of research effort now in using pigs, which have similar metabolism to ourselves and similar sized organs, to see if we can modify them so that we would not reject, shall we say, a pig heart, modifying their immune systems genetically. And again, this will raise, for some people, the idea that we're carrying commodification just that bit too far, and other people would say, well, eat the bacon. So there are questions on both sides. And these are difficult questions to think about, whether it's humans, whether it's future humans, whether it's animals, that people will arrive at different conclusions, thinking equally from an equally Christian perspective, or Muslim perspective, or whatever it happens to be. So, that's all for now, but watch this space.
Great. Provoke some questions. Over to you. Yes.
The sea ships, does that mean we'll have eugenic Olympics?
Well, there are some forms of genetic modification that will be very, very hard to detect. So if you made an embryo that was producing more erythropoietin, one of the essential blood proteins, or more hemoglobin, by doing it genetically rather than by blood injection, you would find that very hard to detect. And there's a lecturer in sociology at Paisley University who's written a book called Genetically Modified Athletes. And his view is that we're not going to be able to stop this, so let's let it go. And that means the countries with the best genetic technology win the Olympics. But it's not a facetious question at all. It's actually out there in discussion. And this is why I threw football in. So, can I actually get Palace back into the Premiership? Instead of lurking at the bottom of the championship. That would be big ask, would it? Probably be. Yes.
Yeah.
Last night in your talk, you talked about the dark side of evolution
Yes.
and things like that, and part of that being genetic disease. But I don't know if you want to talk a little bit about some of the recessive diseases where you have 2 copies, you get the disease like sickle cell anemia, but research shows that with 1 copy you're actually protecting against malaria, for example. So it's beneficial until they come together.
Absolutely. This is called heterozygote advantage, where these genes were actually 1 copy of the gene does have some small effect on you, and with sickle cell anemia, the red blood cells are slightly affected, but they're affected enough so that malaria parasite can't get in. So if you live in a malaria area, then you are resistant, which is, in gene ecology terms, i.e. the ecology of the occurrence of genes, it is believed that is the reason why that mutation has been maintained at a higher level in the population than we'd otherwise expect. Because once we start getting cures and vaccines for malaria, that heterozygote advantage will disappear. Cystic fibrosis has also caused Parkinson's because the carrier frequency is still very high. And we wonder why that is. And some people believe that the heterozygote advantage is that it gives you some sort of resistance to cholera. And that makes sense when you understand what the genetic mutation actually does. It does relate to water relations and salts in the gut as well as the lungs. So that is a possibility. That's just going to hang you around. So heterozygote advantage people, for some genes only, if you've got 140 copy, there's enough effect for it to have an advantageous effect on you.
Back, yes.
Have you seen an old movie called Gattaca?
Yeah, Gattaca it's a very interesting film. I mean, you know, it gets widely shown now in schools for 16-year-olds, 17-year-olds doing their, as we say, their AS or their A2. Gattaca, if you don't know the plot, is a society in which most couples elect to have IVF, and therefore their embryos are pre-selected to be valid. So these are the valids. But there are couples living in more primitive surroundings, or who, like the hero in the film, who was a result of a little escapade in the back of the parents' car when they were students, who are born without IVF, and so they're born normally, but they're all given a complete barcode genetic test at birth to see if they are also valids. And the hero is not valid. He has a gene that predisposes him to a severe heart condition. And he tries to fool the authorities to get onto the space program. It's a fascinating film. So the key thing there is that is a barcode baby, and the government asked the 2 committees that look after the ethics and the practice of genetic testing in Britain to see whether we should do that for all newborns, not just the 5 things, but a complete barcode of every possible genetic condition. And you know, I have ethical worries about that. For whom is that information intended? But the 2 committees came up with the idea that no, we shouldn't do it because it's too expensive. and the costs didn't outweigh the benefits. But yeah, Gattaca, go and see, get the DVD, it's very good.
My favorite scene is him crawling up a spiral staircase.
Yeah, because, well, it's a helical staircase, isn't it? It's a single helix.
Yes.
Just following up on that, isn't that, you said they said, we won't do it because it's too expensive, but presumably there'll come a time when it isn't too expensive.
Yes,
But isn't that the whole problem about the stop button, that actually once you can do it, eventually, especially if we're thinking centuries or millennia, it's going to be done, isn't it?
We can't
All
undo
we
it.
can do is break it, slow it down maybe. Is that fair?
I think that's true. And this is why I think genetic technology is a slippery slope. The boundaries are very hard to define. In much ethical decision making, you reach a fence where it's very clear that actually, you know, that most thinking people would not go any further than that. But with genetics, you know, there is no clear barrier. So I think you're right. That maybe in 20 years' time, it will happen that all babies are barcoded with the best possible intentions. I was nearly a Keith Everett there. Best possible taste. You know, you've got a medical prehistory that enables parents and clinicians to manage the child and to look after it properly. But you know, the insurance companies will want those data. employers will want those data.
Yes.
What will societies do where even now they want to have male babies?
Well, interestingly, pre-implantation genetic diagnosis and, of course, prenatal screening will both give you a clear idea of whether the embryo or the fetus is male or female. In Britain, it is forbidden to use genetic selection for having babies of a particular sex, unless there is a very, very strong medical reason. You know, the boys in this family are bound to have muscular dystrophy. That would be the only reason they would do it. But it feels that in societies where little girls are left out on the hillside, it is still cheaper to leave a little girl on the hillside than to do the genetic test. But as I say, you're not allowed to, in this country to test genetically, just have a boy or just have a girl.
Some of these things you're talking about, you mentioned sort of blue-eyed babies and all the rest of it. Well, it's an awful lot to go through just for...
Absolutely. That's why I say it's not mass technology. You know, that selecting for blue eyes firstly involves you having IVF. And the best clinics in the world are not getting more than about 25% success rate. And then, of course, you may go through the whole heartache if there aren't any blue-eyed kids. And the same is true, incidentally, of pre-implantation genetic diagnosis for serious conditions. It may well be that you go through 1, 2, 3 cycles. You get the embryos free of the condition, but the pregnancies doesn't take. And that happens. And in Britain, the NHS will only pay for 2 cycles of IVF, even if there's a genetic condition involved. So yeah, so you need to be determined, and I think Gregory Stock makes that point in one of his books, you know, that we should have a free-for-all, but these are going to be they must really want to do this, to put the woman through that. And I think maybe too many males have been writing here.
So that might be why you would have the correction of faulty genes in a fetus, because the IVF was too difficult.
Yes, it would be quite difficult to do in a fetus. You'd have to wait till the child was born. So these instances we've been talking about, the severe combined immunodeficiency, the SCID, the genetic therapy is generally done when the child is about 2, so they've had to be very cosseted and looked after by then. Cystic fibrosis is done as soon as possible. The other condition I talked about, congenital blindness for genetic therapy, was done actually on a teenage boy in the USA and a man in his 30s in Britain.
So what about this germline gene therapy?
Germline gene therapy, well, you're suggesting that people will try to... For germline gene therapy, you need to handle the egg, not the fetus. And so you're still in IVF country.
So don't we need more of a conversation about what it means to be a person?
Yeah, and John, I've deliberately not trodden on that ground because John will talk about it. But I will say that there's a lot of debate in Christian and in other circles about, you know, do we regard those early embryos as people? And this is an area where I get very cross when some Christian might say, this is the Christian position. They are people. And I get cross because there are many thinking Christians who do not accept that viewpoint. And I'm not going to tell you what mine is, but you know, there isn't a Christian position on this. And you can have a lot of debate. But that would be a focus for some of the debate. Is it okay to discard embryos? Is it okay to discard embryos even if we know they're going to be very, very ill if they're born as children?
Yes.
This is all of the sort of developing the ethical issues with sort of treating something like SCID. Okay, we can treat this child, but then that child grows up and has children of their own,
and has a 50%
passing on the problem.
Has a 50% chance.
Okay, 50% chance. Okay, but are we not then almost increasing or perpetuating the problem for the next generation? Which raises a huge, you know, another ethical issue.
Well, that raises a societal ethical issue, but I now want to take you to the family. It's got a little boy who will die because they will never ever be able to protect him from all the viruses and bacteria that pass. And so you've got familial and social ethics weighing against society's social ethics.
I wasn't saying it was easy, but you know, it immediately raises that issue.
It does mean that that, as I say, when the little boy becomes an adult, he has a 50% chance of passing on the faulty gene because his genome has still got the gene in it.
I mean, arguably, you think, well, okay, in 20 or 30 years, we may be able to fix that permanently, so it's worth the risk.
Yes, yes. That would be my view.
But you have to deal with the person in front of you.
Absolutely, you deal with the family in front of you, yeah. What is the neighbourly, virtuous thing for a Christian to do? Who is my neighbour?
Yes, you
So, just the other side of what it means to be a person. One is experimenting on embryos and all that side, but another is if you take it further and further down the line towards from therapy to enhancement to beyond, unless we have that debate, what being human means,
Yes.
could we be moving towards making something that is in some way superhuman and is no longer recognizable as the human with vulnerabilities that we accept as part of just life and a good part of life?
I'm contracted to write a book for Lion Hudson, which we're tentatively calling Superhuman or Post-Human.
Right. Yeah, exactly. Very interesting.
And yes, we're getting out into this area of enhancing, deliberately enhancing individuals, quite deliberately. And the Transhumanist Society, which is quite a strong movement, have as a strapline, better than well. And they're thinking about developing, if you like, a small superclass of people whose cognitive abilities are enhanced, maybe by interfacing with IT devices, maybe by drug taking. I find it interesting that many parents in the USA, where you can buy Ritalin over the counter, in order to increase the concentration span of their teenagers when they haven't got ADHD. You know, these are just normal teenagers. So there's obviously a will out there to have that sort of enhancement, and that's another thing Madeline Bunting discusses in her article. So it will be a small group of people who have deliberately enhanced themselves in order that they can be superior in cognitive ability, maybe in physical abilities, than their peers. And I think that, for me, is much, much more of where the debate needs to lie. What does it mean to be human? With our frailties, our susceptibilities, as we are, you know, short or tall in stature, etc., as we are.
Yes, yeah.
Does this mean that in future generations, people who are born dwarfs or with any other abnormality will be considered third-rate citizens
Well,
and despised?
sadly, they already are. There is discrimination against people who don't fit in with the rest of us. And you raised the issue of dwarfism. It's a very, very relevant one because Professor Shakespeare at Newcastle University, Professor of Medical Ethics, did his research here in Cambridge, and he has achondroplasia, a dominant mutation that gives you short stature, usually slightly enlarged skulls and so on. And there's a lot of discrimination against people with short stature. But he was obviously making a career for himself in sociological research. And he and his wife started their family when they were here in Cambridge. And the genetic test for achondroplasia wasn't available. It is now. But the scans showed that their first child had achondroplasia, the early scan. And the advice from the clinician, talking to a father who was clearly was to abort the fetus. And there's a beautiful moving, although one or two cases biologically inaccurate, film that Channel 4 made called Ivy's Genes. Ivy is the little girl. She's now about 17. I mean, it was astonishing that they should be so insensitive. But so there is that. And there's a lot of discussion in the film called Ivy's Genes. If Channel 4 have it on available, it was made back in the 90s. If that's available, there's some lovely discussion about discrimination against people who have some sort of difference from ourselves. So, and I think one of the worries about genetic selection is that our idea of normality may get squeezed. And this is why the HFEA is taking such a strong line on what should be tested for and what shouldn't. Because conditions that are manageable in life and people leading normal, fulfilled lives are not allowed to be tested for phenylketonuria, achondroplasia. Only things that produce very severe medical conditions are allowed. But the idea that we will squeeze out the normality is actually quite worrying. You know, the kind of normal distribution curve gets narrower and narrower, and people therefore suffer discrimination. Dave's been waving his arm back.
I think Dave at the back.
So, that's what it's called, personality are all part of DNA.
No, they're not all part of DNA. I mean, genes do affect our personality. I'm going to put it that way around.
Yeah.
Our genetics has some effect on our being in terms of personality and behaviour, but only some effect.
So you couldn't sort of like eradicate, lack of patience in your DNA.
Well, we do know of a certain one particular genetic change that predisposes people to violent reactions, emotionally violent reactions. But again, I have to emphasise this is a predisposition to it. It's not fixed. We know of chemical conductors in the brain that make people more predisposed to the state of depression. Again, it's a predisposition. Those are built into our DNA, but they don't make you automatically depressive or violent. So that's the sort of level we're talking about. So, the idea that we might produce a really cheerful, whatever it happens to be.
Dave, you had one.
Without stepping on toes of this afternoon's lecture, I think you've already said this really does come down to what does it mean to be human. And it seems to me we've got an increasing divide between a view that's based on an atheistic version of evolutionary biology that says we are about self-improvement and we are about becoming better, more evolved and redesigning ourselves. And another view, which is not only Christian, but brings in a moral dimension that says we have responsibilities towards the weaker, towards the vulnerable, towards those who are not going to come out top of the pile simply by natural selection. Now, if we take that moral view, surely part of being truly moral beings is taking care for those who cannot give any form of consent and any morality and make any moral decisions for themselves, and that includes animals. Now, I've just been reading Andrew Lindsay,
yes, of course,
latest book on this, and he makes some very, very interesting comments about the way we dehumanize ourselves in the way that we treat animals, in research, in seeing them simply as there for our pleasure, in using them simply to grow organs for ourselves and whatever, lots of other things. So, some very interesting moral dimensions there.
I don't go all the way with Lindsay, but my own feeling about animals is that the Christian community has not thought enough about this topic. And when I wrote my book, Life in Our Hands, I introduced some discussion of Andrew Lindsay and of other views within the Christian community. And the publisher said, do you think this should be in the book? And I said, yes. I had a number of arguments with that particular publisher for that particular book, but that was one of them. And I think we do need to think about that. It is all about interaction with the rest of the natural world. I mean the rest of the natural world because in some senses we're part of it.
Yeah.
Yes. Looking at animals once more, what are the other animals that have got very scarce, I don't know, say a group of tigers in an isolated forest in India with a very limited gene pool? How could they survive? What is the sort of tipping point whereby the sort of pool of healthy genes is not there anymore?
Yes. You're talking about something we call genetic erosion. Genetic erosion is when the population gets so small that the amount of genetic diversity becomes threatening. And that number will vary according to the species. I mean, species with simple genomes can actually find enough to get away with it. I don't know what the critical number will be for a tiger, but some birds have been rescued from very, very low numbers. And I think what will happen there is that the low numbers that survive are the ones, if you like, whose genotype makes them more able to survive. But genetic erosion is a serious problem in conservation, and you hear mining companies magnanimously saying, "Yeah, we're going to keep a small amount of forest," but that doesn't work.
Yes.
How important is it in the future for politicians and democracy regarding what we're talking about and our feelings about what part of the world we might be living in when we put people in power and we have these fears of what we might be up to.
Well, we do live in a political world, and I alluded to that yesterday, and I'll give you a specific example. It matters not what you think about the ethics of embryonic stem cell research. There are at least two views of the Christian community, probably more. It matters not where you lie, but what is very clear is that the 2001 modification of the HFE Act was pushed through Parliament by very, very highly motivated politicians led by Dr. Ian Gibson and Dr. Evan Harris, because they wanted to see Britain retain its lead. They clearly didn't know what's going on in the USA, where it's rather what's going on. They wanted to see Britain retaining its lead in Europe in this research. And so, in my view, and this again doesn't tell you what I think about stem cells, but in my view, the modification of the Act was actually pushed through Parliament by strong lobbying from that particular group who form part of the parliamentary committee. So politics does have a role to play in the way society will adopt or not adopt these technologies.
Interesting that neither of them are MPs anymore, and Evan Harris particularly with his defeat.
Yes, it was quite close. I'm
By
sorry to
a
say
Christian from St Aldais Church.
Yes, I know. You know, one was torn by my party politics would say I'm sad about Evan's demise.
Ethically,
but I've had some discussions with him when he talked about people who oppose scientific evidence as being Luddites, and I said, no, I mean they're not Luddites. I speak to some of these people; they're thinking people. They just disagree with you.
Indeed. Yes.
And there's hand back after that.
Yes.
Yes. A couple. You may have answered this. Do we know why genes mutate in the first place?
Yeah. Okay. So
just a quick another one. And how this whole epigenetics has quite informed your view of what makes us up as a person when we think of the biblical kind of body, mind, spirit. I don't know whether that's informed your thinking about that.
Okay. What causes mutation is when the DNA is being copied. Every now and then, it's a very rare event. The wrong building block is inserted, and that will change the makeup of that gene. Now, we have correction mechanisms. They've evolved alongside our genes, and probably 99 out of 100 of those events get corrected. And I think we've calculated that for animals like ourselves, that it's something like 1 in 10 to the 11 DNA replication events will introduce a mutation, a change. You think how many cells we've got. So it's caused by if you like, a biochemical aberration when the wrong base is inserted, the wrong building block is inserted, and the correction mechanism doesn't correct it. So they are very rare events. That's one form of mutation. Another form of mutation is where the replication mechanism skips a bit of the DNA, so you get a gap. And we've talked about this insertion. These are very, very rare events, but in terms of the number of generations and the number of cells in each generation, then they are frequent enough for a mutation to occur from time to time. And in terms of does it inform me? I mean, there's a level of our existence that I can experience, and that's called spirituality. And I can experience that, I can feel it, and it has an effect on me, but I can't put that down to my genes. You won't find the genes for the spirit anywhere. No, just as you won't find a location in the brain for the spirit. And I'm glad we've got Alistair coming to talk to us this afternoon about the brain and spiritual experience. So genetics informs me of how my system works. It doesn't say very much about me as the whole holistic person, the influences that have come from my family, from my background, from all this other stuff, from God, from the indwelling of the Holy Spirit, that stuff.
I think we'll make yours the last one.
Back to the animals,
Yeah.
and leaving aside some of the ethical issues, using genetic modification to create organs to transplant into humans, what's being done about the possibilities that opens up for the migration of animal diseases?
Yes, absolutely, absolutely. This would be, we're looking to risk benefit analysis. Somebody is going to die of a liver failure. but we put a pig liver in, which is beautifully compatible with that recipient, but viruses that have lain dormant in the pig genome might get transmitted into that particular human. We call them zoonoses, diseases of animal origin, and it is a risk-benefit analysis. And until somebody starts doing it, we will not know. Mm-hmm.
Thank you. Well, that's absolutely terrific, John. Thank you very much.